Welcome to section 3.6, Active Transport. In this section, we'll explore how cells use energy to move substances against their natural concentration gradients, an essential part of how cells maintain internal balance and perform specialized functions like nerve signaling and nutrient absorption. Our goals in this section are to understand how electrochemical gradients affect ions, describe various forms of endocytosis, phagocytosis, pinocytosis, and receptor-mediated endocytosis, and to explain the process and purpose of exocytosis. These concepts help us understand not only how individual cells function, but also how systems like the nervous and immune systems operate. Electrochemical gradients form when there are differences in both ion concentration and electrical charge across a membrane. These gradients drive ion movement and are vital to functions like nerve signal transmission and muscle contraction. Think of them like a battery, charged and ready to power the next biological task. The sodium-potassium pump is a cornerstone of active transport. It uses energy from ATP to move 3 sodium ions out of the cell and 2 potassium ions in. This process maintains a membrane potential, which is essential for electrical signaling in neurons and muscle cells. It's one of the clearest examples of primary active transport. Active transport requires energy, usually in the form of ATP, because it moves substances against their concentration gradients. This is especially important in places like the digestive tract or root systems in plants, where cells must absorb nutrients that are more concentrated inside the cell than outside. There are two main types of active transport. Primary active transport uses ATP directly, like the sodium potassium pump. Secondary active transport, or co-transport, uses the energy stored in existing ion gradients. For example, the sodium glucose symporter brings glucose into intestinal cells by leveraging the sodium gradient created by the pump. Endocytosis is the process of bringing materials into the cell. The plasma membrane folds inward and encloses the material in a vesicle. This is how cells internalize nutrients, remove pathogens, and regulate surface proteins. Phagocytosis is cell eating, used by immune cells to engulf bacteria and debris. Pinocytosis is cell drinking, taking in extracellular fluid and the molecules within it. Receptor-mediated endocytosis is more selective, where specific molecules bind to surface receptors before being brought into the cell. A breakdown in this process can lead to diseases like familial hypercholesterolemia. Exocytosis is how cells expel materials. Vesicles containing substances like neurotransmitters or hormones fuse with the cell membrane to release their contents. This is critical for neuron communication and endocrine signaling. It's also how cells dispose of waste and recycle membrane components. Active transport allows cells to move substances against gradients using energy. Electrochemical gradients power essential functions, and specialized processes like endocytosis and exocytosis enable cells to manage their internal and external environments. Together, these systems help cells stay alive, respond to changes, and communicate with the world around them.